Let's review the Unit 7 of AP Biology on evolution in about 20 minutes. Hey guys, this is Mikey with Avo Prep Academy, and on this channel, we review AP Biology content. In today's video, we'll be cramming all the content covered on the second last unit of AP Biology, Unit 7 on evolution and its mechanisms. This unit largely covers chapters 22, 23, and 24 of Campbell Biology, focusing on Darwinian selection, microevolution, and macroevolution. And it dives also a little bit into phylogenetics from chapter 26. Let's do a review in essentially that order. We begin this unit by studying a bit of history leading up to Darwin's publication of The Origin of Species in 1859. But to put everything into perspective, let's first put some things on the table first. See, the two main ideas that the chapter establishes is that prior to Darwin's proposal of his theory of evolution by natural selection, there were two major prevailing thoughts common among scientists and laymen alike. First is the fixedness of species, in that most people believe that all the species in the world have always been and will always be. No speciation, no extinction, just fixed set of species. Second is the young Earth, with most people believing that our planet was just about 6,000 years old. Of course, we now know that our planet is close to 5 billion years old. The history of breaking away from these ideas would lead to Darwin's development of his theory, and we begin in ancient Greece with Aristotle. Now, the chapter begins here just to mention that Aristotle, while a great philosopher, believed that all species were fixed. Then, skipping quite a few centuries later, we even see that even Carolus Linnaeus, the scientist responsible for the binomial nomenclature system, would pretty much be on the same page as Aristotle, believing that a higher power had individually designed each and every species which he was attempted to categorize. But it was around then that the idea of fixedness in species began to come into question. The main issue was fossils, with discoveries of ancient fauna and flora that resemble nothing that exists in our modern times. So now that the fixedness of species was becoming questionable, we can link this very idea to the challenges against the young Earth hypothesis. And that's because these fossils were discovered in sedimentary rocks. The strata within them, or these large segments of distinct rock formations, would have taken millions, if not billions, of years to form. But here too, we see some resistance by people like Georges Cuvier, who argued that the strata that we see have been formed within a short period of time because the early days of our young Earth were filled with catastrophes like the Great Flood. This idea, which he called catastrophism, would be successfully challenged and usurped by James Hutton and Charles Lyell with their theory of uniformitarianism. In Principles of Geology, Lyell would argue for this theory, claiming that the gradual changes we observe today occurred at the same rate in the past, directly undermining catastrophism while simultaneously insinuating that our planet was, in fact, very, very old. So at this point in the early 1800s, the science of natural history is on somewhat shaky grounds. No longer could we believe that the species were fixed, nor could we believe that Earth was quite young either. And here we begin to see thinkers leveraging both this newfound timeline in Earth's history and the idea of changing species. The man I'm thinking of isn't Darwin, but rather Jean-Baptiste Lamarck. Now, just to put it out there, Lamarck was wrong, but what he tried to do is so important. See, Lamarck tried to explain how species could change through naturalistic mechanisms rather than supernatural or directed by mankind, like in the case of artificial selection, which we have become pretty good at by this point. In his theory of acquired inheritance, often called Lamarckian evolution, he proposed that organisms change through time due to use and disuse of body parts in its lifetime. For instance, a giraffe who stretched its neck throughout its life to reach a higher leaf on a tree would give birth to giraffes with slightly longer necks. Now, while it isn't necessarily true that what happens in your lifetime can impact your offspring, Lamarck's proposal began seeding ideas among scientists that, first, species could change, and second, there potentially existed a natural mechanism that could explain how that change occurred. And finally, we get to Darwin. We learn about Darwin's role as a naturalist on the HMS Beagle and how he made tremendous number of observations on the Galapagos Islands. The main thing to remember here is that one of Darwin's major curiosities was the origin of adaptations, which are features of organisms that allow them to survive and thrive in their respective environments. For instance, Darwin observed that finches living on different islands had very different looking beaks. Some had deep beaks that could crack open seas that were more common in their environment, while others had sharp tweezer-like beaks that could help the birds hone in on and capture insects that they relied on in their environment. And in this picture, we see how Darwin began to think about the existence of not only adaptations, but how species that appear similar may have arisen from ancestors, almost like cousins or some distant family members who share certain characteristics but not others. So at this point, let's flesh out the details of Darwin's ideas about how, over long periods of time, organisms could change to diverge into multiple species from an ancestral one. Now, Darwin made several observations that will help establish this framework. 1. Members of a population often vary greatly in their traits. 2. Traits are inherited from parents to offspring. 3. All species are capable of producing more offspring than the environment can support. And 4. Owing to lack of food or other resources, many of these offspring do not survive. Now, all of these observations make sense. By Darwin's time, we knew how to breed certain traits into both animals and plants. Of course, this would only be possible through the existence of variations and the fact that traits were heritable. On the other hand, observations 3 and 4 seem to have been influenced by Thomas Malthus, who offered a similar explanation for the strife we observed in humanity, such as famine or war, owing much to the overpopulation problem and the finitude of our resources. But it was in fact the genius of Darwin that connected the dots in drawing up the following inferences. 1. Individuals whose inherited traits give them a higher probability of surviving and reproducing in a given environment tend to leave more offspring than other individuals. 2. This unequal ability of individuals to survive and reproduce will lead to the accumulation of favorable traits in the population over generations. To put this simply, Darwin exposed that we were all blinded by something called survivorship bias. This meant that while most people were simply looking at adaptations as part of some grand plan by the universe, Darwin's idea was that the only reason we see adaptations at all is that the poorly adapted individuals never survived to be part of this picture. Take the finches, for instance. Supposing that we begin with a population of finches with some average-looking beaks, given that the environment only offered hard seeds as the main food source, finches with thicker or deeper beaks would have been able to access more food and thereby more energy to lay more eggs than smaller beak finches who never survived nor reproduced. Given that shape and size of beaks are heritable, this would result in more and more finches being born to finches with bigger beaks, resulting in the whole population beginning to look more and more fit to that particular environment. I'm going to jump ahead here a little bit and add something that we typically see a bit later. I want to briefly mention that as selection occurs, it can drive 3 types of patterns. Directional selection selects for one end of the phenotype spectrum, like bigger beaks being preferred over smaller beaks. Disruptive selection selects for extremes and selects against the mean, like having really dark or really light moths doing better than the average colored ones. And finally, stabilizing selection, where the mean is better suited than the extremes. And the common example used here for the exam is clutch sizes of birds, where having too few eggs or too many eggs would both be bad. But now that we have natural selection out of the way, the rest of the chapter focuses primarily on a mountain of evidence that support evolution with some specific vocabulary that you need to know. First, we see evidence of evolution in real time. This is in reference to the development of antibiotic-resistant bacteria. Here, we see how strains like MRSA or drug-resistant tuberculosis have become a growing concern. But there's something really important about natural selection that the course wants you to note in this particular section, and it's that bacteria do not suddenly develop resistance genes when exposed to antibiotics. The way that it actually works is that even prior to the application of antibiotics, there must exist some random mutant bacterial cells that were already resistant to the antibiotics and simply happened to be positively selected with the application of said antibiotics. This is to drive home the point that evolution isn't reactive and merely selects for variations that happens to be beneficial at that moment in time. We also have homologous structures. These are structures in different species that appear similar in their organization due to a common ancestry. Arms of humans, cats, whales, and bats, all with their humerus, radius, and ulna, seem to suggest that the ancestor of tetrapods likely passed this particular arrangement down to these species. And that will be true even if the final usage and the function of these forelimbs may differ between these species. Now, comparative embryology of species shows similarities in early development, again owing to a common ancestry. And adding to this, we have vestigial structures, which are structures that no longer serve a useful function today, but still may exist due to some evolutionary history. Examples would include things like the tailbones in humans, where we no longer have tails, but the bone still exists. And we also see another important term here, which is convergent evolution. Now, convergent evolution doesn't actually refer to species converging back together, but rather certain patterns of anatomy that develop due to similar environmental constraints and the natural selection process that leads to it. These structures are often referred to as analogous structures and include things like the flaps of sugar gliders and flying squirrels and the dorsal fins in whales and sharks whose ancestors never had these traits. And we say that these organisms independently evolved these adaptations. Just keep in mind that what we're trying to support here is the idea that natural selection does, in fact, operate, and that it could explain natural development of adaptations in somewhat of a predictive way. With that, we get to move on to chapter 23, looking closer at how we analyze evolution occurring at the micro level. This part of the unit is devoted to microevolution, which incidentally includes natural selection that we just finished describing. But before we get to all that, we see a slightly more in-depth discussion of what makes evolution work, which is variation. More specifically, we want to focus on the source of variation in biology. Now, considering that most ancestral organisms were likely asexually reproducing prokaryotic cells, the standard replication mechanism should only produce clones. But here we learn that in all of biology, in both bacteria and complex eukaryotic organisms, mutations is the first and foremost source of variation. Mutations, while deleterious in most cases, does often create changes to the genetic data that is at worst neutral and at best beneficial in certain cases. These are now subject to positive selection, allowing the propagation of that new mutant allele within the population. However, we also learn that in sexually reproducing organisms, even more variation can arise from mixing and matching of traits with each generation, with meiosis, random fertilization, and independent assortment playing a major role. So while mutations create new alleles, sexual selection can recombine those alleles in individuals to create almost an infinite number of unique individuals within populations. But now that variation is out of the way, let's get started on microevolution more properly. And we'll do this by creating a formal definition of microevolution. Now, microevolution is defined as a change in allele frequencies across time or generation. So one major step in understanding this concept is to figure out what allele frequencies are exactly. And for this part, let's take a look at an example. In this picture, we have a population of rabbits that have two alleles that contribute to their fur coat color. Genotypes big F and big F for brown rabbits, big F and little f for light brown, and perhaps little f and little f for white rabbits. As we look at this single gene called F, we can determine the allele frequencies by counting the number of big F alleles and dividing by the total number of alleles, which in this case is 20, as there are 10 individuals who each have two alleles. So as you can see, with 13 big F alleles in a pool of 20, we can calculate the allele frequency of the big F allele as 0.65. The little f allele should be 7 out of 20, which can be calculated independently as 0.35, or you could even simply subtract 0.65 from 1 to get that exact same answer. So that's all good for time 0, but as we defined earlier, microevolution is any change to that frequency that occurs over time, and it turns out that in our universe, there are 5 major mechanisms that drive this change. 1. Mutation. 2. Natural selection. 3. Gene flow. 4. Genetic drift. And 5. Sexual selection. Mutation is simple to understand. Of course, if any little f became a big one, or vice versa, in a germline cell of one of these rabbits, then the frequency of these alleles in this population, as they pertain to what is heritable to the next generation, would be changed. Natural selection? Well, that too could be easily modeled. Supposing that we have a snowy winter where the ground turns white, the advantages conferred to white rabbits would allow more of the little f carrying individuals to survive and reproduce, thereby increasing its frequency while simultaneously decreasing the big F allele frequency. Gene flow also makes sense. Given that this population is part of a larger group of populations of rabbits, any rabbits entering or leaving this population would affect the allele frequencies, as individuals moving in and out would naturally bring in or take away certain alleles at this gene F. Now, genetic drift is a bit more trickier. Here, what we see is a statistical effect called a sampling bias, where a large decrease in the population size of this rabbit population could drastically change its allele frequencies. Supposing that by chance all of the rabbits on this side of the population died, maybe due to a volcanic eruption or something like that, then the only survivors would have the big F allele. The example that I've just described is referred to as the bottleneck effect, to describe the thin neck of a bottle through which only a small portion of the population survives to the next generation. Keep in mind that there is yet another effect called a founder effect, where a small portion of the population moves away from the original area to a new area to start a new population population where you would witness the same drastic change due to the same statistical anomaly. Finally, sexual selection plays a major role in microevolution. Supposing that many sexually reproducing species have choosy females, which they normally do, the selection of males by females can not only select for useful traits, but also arbitrary traits like an exaggerated tail of the peacock. These traits, driven by genes and their alleles, would change over time as females act as the gatekeeper of reproductive success. Now that sums up the 5 mechanisms of microevolution, which you should absolutely keep in mind as you proceed onto macroevolution. But before you do that, there is just one extra thing in this chapter which is critical in solving certain problems on the AP exam, and that is the Hardy-Weinberg equilibrium and its equations. But I do think that you should spend at least 10 minutes or so understanding the concepts and how those questions work, so I'll put a link for a more detailed video down in the description below and at the end of this video so you can brush up on that particular topic which pertains to the math portion of the exam. We finally move on to chapter 24 on macroevolution, which links the ideas discussed in microevolution to the bigger patterns of speciation we observed across longer expanses of time. We begin this chapter by exploring what it means to use the word species. To cut to the chase, in AP Biology, we use the biological species concept, which defines species as a group of organisms that have the potential to mate in nature and produce viable, fertile offspring. This means that interbreeding between individuals must be possible and that the offspring should be able to survive and capable of interbreeding with other members of this said group. This works well for animals mostly, in that dogs are dogs because they can produce puppies that grow up to be dogs, while not being able to mate with a cat on the other hand. But what about some close relatives who could produce offspring, like horses and donkeys? Well, they do successfully mate and produce mules, but mules are sterile, and so they fall short of being categorized as a single species under this particular definition. So it seems to work pretty well, but keep in mind that this definition is heavily dependent on interbreeding, meaning that extinct species or asexually reproducing species would require other definitions to clarify the species-species distinction. Once we know what a species is though, we can begin asking what it is about these interbreeding organisms that allow them to produce viable and fertile offspring. The answer is simply the similarities in their genetic makeup. For instance, humans have 46 chromosomes, and we produce haploid sperm and egg cells that contain 23 each. Each of these haploids can come together, aligning those homologous chromosomes in a way that allows for their correspondence to coordinate gene expression. If the homologous chromosomes contain the same genes at the same loci, the alleles can then work together to produce individuals who express genes properly, which would explain the viability part. And they also produce haploid cells that have the proper number to align with the haploid cells of another individual, which then explains the fertility part. And what keeps genetic makeup consistent within a single group of interbreeding organisms is gene flow. You see, this continuous gene flow among individuals of a population allows for any microevolutionary mechanisms, be it selection, gene flow, or mutations, to continue mixing into the overall picture of their genome. Imagine a paint mixer that continuously homogenizes the paint contained within. As new droplets of colors are added, like a mutation in this analogy, it would be eventually mixed in, allowing the color to shift together but never disparate in different parts of the solution. Then I think we can logically ask the next question, which is what would cause the genetic makeup to sufficiently change between individuals of a population to begin the speciation process? If we kept up with the paint mixer analogy, it would be simply having two mixers with two paint mixtures that never intermixes to become one color. And in terms of biology, restriction of gene flow, otherwise known as reproductive isolation. Here we see two major pathways to speciation: allopatric speciation and sympatric speciation. In allopatric speciation, we see a geographical barrier that cuts off gene flow between what was once a single population. The microevolutionary events occur independently in the two now separated species, resulting in the buildup of sufficient genetic differences over time. Whether it be differences in selectional pressures, chromosomal mutations that alter the structure of the genome, or any of these other microevolutionary mechanisms, all of these small yet cumulative events would occur in complete independence, resulting in further and further divergence over time. But in sympatric speciation, we contend with the idea that a single group of interbreeding organisms suddenly divide into two separate breeding pools, even in the same area. We see several examples in the textbook, with the most notable being habitat differentiation. Here we see that a group of maggot flies that once parasitized the hawthorn trees moving to apple trees that are introduced by European settlers. Because apple trees mature earlier, the flies that are now on these apple trees undergo a different selectional pressure to lay eggs earlier and earlier, eventually reaching sexual maturity at different times against their original hawthorn population counterparts. This temporal isolation and habitat differentiation occurred in a single orchard, resulting in a sympatric speciation event. Now, other examples include things like sexual selection, wherein females can become selective in choosing a particular type of male within a single population, producing sons that look that way and daughters who choose that way as well, subsequently dividing that population into two separately breeding pools. But with all that in mind, we see that speciation results in barriers to reproduction that forms at various levels. With AP biology, we need to know that these barriers exist as prezygotic barriers, or factors that inhibit mating and the formation of a zygote, as well as postzygotic barriers, or barriers that keep species apart even after the zygote had been formed. In prezygotic barriers, we first have habitat isolation, where species occupy different areas of the globe, temporal isolation, where they mate at different times, as well as behavioral isolation, where mating behavior isn't recognized by the opposite member of the sex of another species. These factors are all pretty large scale and can inhibit different species from mating. But even if we have a mating attempt, we have mechanical isolation that physically make it difficult for individuals to copulate, and gametic isolation that inhibits sperm cells from fertilizing the egg, again due to sufficient genetic differences. Now, supposing that two species can actually try and mate and form a zygote, this is where the postzygotic barriers kick in. First, we have reduced hybrid viability, which means that the hybrids simply cannot survive long enough to reproduce, and we have reduced hybrid fertility, which is accounted by how hybrids, like mules for instance, are sterile and cannot reproduce on their own. In yet other species, we even have hybrid breakdown, where hybrids can reproduce but eventually fail at some point later in a number of generations. Now, the last part of this chapter deals with the time course of speciation. Here we have phyletic gradualism, which stipulates that species change slowly over time, versus punctuated equilibrium, which states that species remain fairly same for long periods of time, only to change relatively quickly when speciation events occur. Both are valid and can be seen in our evolutionary history on our planet. So that about sums up the major concepts from this unit. There is yet a bit more to discuss on how to create and interpret phylogenetic trees, but we can dive into that one a little bit more in a different video. Now, don't forget to watch my other video on the Hardy-Weinberg equation to complete your toolkit in cooking this exam at your school and in May for the official AP exam. If you think this video has been helpful to you, be sure to smash that like button and click subscribe. And most importantly, share it with that one kid who always falls asleep in class. This has been Mikey with Avo Prep Academy. See you all very soon.